Good morning, dear students. This is your teacher Neelam Elawat, and we are doing chapter 12 Biotechnology and its applications. In the previous video, dear students, we have talked about the role of biotechnology in agriculture. And today in this video, we'll be talking about the applications of biotechnology in medicine. Okay, that is, what is the role of the biotechnology in medicine? See, the recombinant DNA technology, students, it has an immense impact in the area of the healthcare. Okay, it has immense impact. So basically, this technique helps in the mass production of safe and more effective drugs. So this is the first one. That is because of with the help of the biotechnology, we can produce the more safer and more effective drugs. Secondly, this technique also does not induce the unwanted immunological response as has been observed in case of similar products that have been obtained from the non-human resources like the animals. See, whenever a particular substance is induced in the body, then if it is not identified as self-defense mechanism, then our immune system will start acting against it. So that will bring about the immunological response. So two things. First of all, it will help in mass production of safer and effective drugs. And secondly, it will not trigger the unwanted immunological responses in the body. So if we talk about the present situation, at present there are 30 drugs that have been produced with the help of the recombinant DNA technology, and out of those 30, 12 are being marketed in India. So just imagine, out of 30, 12 drugs they are being marketed in India alone. So I'm here showing you a picture. Just look at this picture magnified. This drug has been prepared by using the biotechnological applications. Okay, this is one of the drugs that is being utilized in India. So let's talk about, I'll be talking about the three examples here. First is the genetically engineered insulin. Okay. Second is gene therapy, and the third is a molecular diagnosis. Means how does it helps in the identification of particular disease. So let's start with the first, that is genetically engineered insulin. See, we know, students, that insulin is a hormone which lowers your blood sugar level. Okay, so it is being secreted by the pancreas in our body, right? So we know that in humans we have pancreas. Humans में क्या है, आपके pancreas होते हैं. This is an organ in the body, and these pancreas, they produce a hormone which is named as insulin, and this insulin, it reduces your blood sugar level. Okay, it reduces sugar level. The blood sugar level is reduced in the blood. So in this case, the insulin plays a very important role. So the persons in which either the insulin is not secreted or the pancreas are damaged so that they cannot produce insulin, so the blood sugar level will increase, and in this condition the patient will have a diseased condition which is called as diabetes. So if the insulin is not able to manage the blood sugar level, then in that case they will exhibit the disease diabetes. Okay, so that is why we say that insulin is used to manage the adult onset of diabetes. So insulin, it controls your blood sugar level. That means it reduces the chances of being diabetic. Now initially the patients that had diabetes, जिन लोगों को diabetes थी, they were given the insulin that is being obtained from the animal resources. तो उनकी बॉडी में जो है इंसुलिन नहीं बन पा रहा था, या इंसुलिन कम अमाउंट में सिक्रीट हो रहा था. सो इन दैट केस the insulin which is produced in different animals, that was extracted and it was to be induced in their body. So initially there were two organisms, that is cattle and the pigs. Now these animals, they were utilized. Basically when these animals, they were slaughtered. Okay, आपको पता है ना कि बुचर खाना होता है, उसके अंदर आपके कैटल और पिग्स को स्लॉटर किया जाता है, मारा जाता है, किल किया जाता है. So when these animals, they were slaughtered, So from their pancreas, this particular protein was secreted. Insulin was obtained from their bodies. But this insulin, it used to trigger the immune system. So there were certain reactions that the body used to face. तो जब इन रिसोर्सेज से इम्यून सिस्टम जो है इन रिसोर्सेज से इंसुलिन प्रोड्यूस करके बॉडी में इंड्यूस किया जाता था ह्यूमन बॉडी में, सो इट वाज ऑब्जर्व दैट इट ट्रिगर्स सम काइंड ऑफ एलर्जी एंड अदर टाइप ऑफ रिएक्शंस. तो बॉडी में इसकी वजह से कुछ एलर्जिक रिएक्शंस हो जाते थे. बट नाउअडेज इट इज़ पॉसिबल दैट वी कैन प्रोड्यूस ह्यूमन इंसुलिन बाय यूजिंग द बैक्टीरियाज. आज की डेट में हम बैक्टीरियाज को यूज़ करके ह्यूमन इंसुलिन प्रोड्यूस कर सकते हैं. How that process is done, let's understand that. See, we know this is a human cell. In the human cell, nucleus is present. In the nucleus, there is chromosomes are present. Okay. Now these chromosomes, or you can say DNA replication will occur. Then translation will occur. mRNA will be produced. Okay, mRNA will be produced. Functional mRNA. From this functional mRNA, we will extract the gene which is responsible for the production of insulin. सो हम mRNA से जो जीन है जो इंसुलिन प्रोडक्शन के लिए रिक्वायर्ड है उस जीन को आइसोलेट करेंगे. नाउ वंस दिस जीन इज आइसोलेटेड दिस जीन इज ट्रांसफर्ड इनटू द प्लास्मिड. ओके, बैक्टीरियल प्लास्मिड. इस जीन को हम बैक्टीरियल प्लास्मिड के अंदर इंसर्ट कर देंगे. नाउ दिस प्लास्मिड इज इंसर्टेड इनटू द बैक्टीरिया. And now what will happen, when this bacteria will multiply, automatically this plasmid will also multiply and the gene will also get multiplied and there is a production of the insulin. And then this insulin can be extracted from the bacteria. It can be supplied into the human body. So this is how the process is being done today. Now the hormone insulin that is being produced, it basically consists of two पॉलीपेप्टाइड चेन्स. जो आपका हार्मोन है इंसुलिन उसके अंदर दो पॉलीपेप्टाइड चेन्स होती हैं. देयर इज चेन ए एंड चेन बी. जस्ट लुक एट द डायग्राम. दिस इज योर चेन ए विच इज इंडिकेटेड विद ब्लू एंड द सेकेंड दैट विच इज इंडिकेटेड विद येलो is a chain B. And these two chains, they are linked with each other by disulfide bonds. ये दोनों जो है पेप्टाइड बॉन्ड्स जो चेन्स हैं they are linked with each other by disulfide bonds. So insulin hormone consists of two chains, two polypeptide chains, chain A and chain B, and those two chains they are linked with each other by polypeptide bridges, polypeptide linkage. Okay. Now in mammals basically the insulin is produced as a prohormone. This means when insulin is synthesized by the pancreas, it is in inactive state. Okay. जब पैनक्रियाज जो है वो इंसुलिन बनाते हैं वो इनएक्टिव स्टेट में होती है और इसीलिए हम उसको प्रो हार्मोन कहते हैं. एंड देन इट इज़ कन्वर्टेड इनटू अ फंक्शनल इंसुलिन. सो लेट्स सी, सपोज देयर इज पैनक्रियाज. नाउ दीज पैनक्रियाज दे विल प्रोड्यूस द inactive insulin, and this inactive insulin will be referred to as prohormone. Okay. Now this inactive insulin will be converted into active insulin. So ये जो inactive insulin है इसको आपका active insulin में convert किया जाएगा और इस process में क्या है, एक strand remove किया जाएगा DNA के अंदर से, that is your C peptide. Okay. So pancreas, when they synthesize insulin, this synthesized insulin is in inactive form. That is why we call it as a prohormone. Now this prohormone, it will be processed so that it can be converted into an active insulin. And when this is converted into active insulin, in this process, there is a C peptide which is to be removed. Let's understand this. So let's look at this diagram. This is the insulin which is secreted in humans. So it is referred to as proinsulin because it contains certain unfunctional parts. So those unfunctional parts, they will be removed. So this proinsulin, it will undergo the processing and it will be converted into insulin. When it is converted from proinsulin into insulin, a C peptide chain is removed from the polyinsulin. ठीक है, प्रोइंसुलिन. प्रो इंसुलिन जो है वो हमारा इनएक्टिव इंसुलिन होता है. इस प्रो इंसुलिन को हम प्रोसेस करते हैं तो हमारा एक्टिव इंसुलिन प्लस हमारी सी पेप्टाइड रिलीज़ हो जाती है. तो सी पेप्टाइड वो एक्स्ट्रा स्ट्रेच है, वो एक्स्ट्रा पार्ट है क्रोमोसोम का जो आप रिमूव करने के बाद आपका इंसुलिन हार्मोन फंक्शनल हो जाता है. Okay, so insulin hormone, it basically consists of two polypeptide chains A and B, and the insulin is released in an inactive form. That is why it is called a prohormone. Then it will undergo the process of amplification. It will be processed and it will be converted in active insulin. And during this process, an extra amount of extra strain of DNA, extra stretch of DNA will be removed. That is referred to as a C peptide. Okay, that is referred to as C peptide. Now initially, if I talk about an example, in 1983, there was a person named Eli Lilly. Okay, in 1983, there was a person named as Eli Lilly. Sorry, an American company named as Eli Lilly. Now this company prepared 2 DNA sequences. Okay, we know that insulin is made up of 2 polypeptide chains A and B. So this company, which is Eli Lilly, it prepares 2 DNA sequences. So like, let's suppose this is one sequence, this is second sequence. So this sequence will produce your chain A of insulin, and this will produce chain B, right? So this American company, it prepared 2 DNA sequences that will correspond to chain A and chain B of insulin. Now these two, see, as I told you that it prepared 2 DNA sequences, A and B. A will be responsible for production of chain A and B will be responsible for production of chain B. Now these two parts of the DNA, they were introduced into the plasmids. अब ये जो DNA sequence है, ये plasmid के अंदर insert कर दी गई है. Okay, they were inserted into the plasmid of DNA, and that will produce your insulin chains. So in the plasmid, the different chains of the insulin, that is chain A, chain B, they will be produced separately. Now these two chains, they will be extracted. Okay, they will be extracted and then they will be combined by using the disulfide bonds to create the human insulin hormone. Okay. So this was the first example where insulin was prepared by utilizing the DNA sequences, by utilizing the E. coli bacteria, right? So we can say with the help of DNA technology, we are able to produce large amount of insulin and by utilizing the bacteria. And it is more suitable in comparison to the insulin which is extracted from the animals. Okay. I hope this is clear to everyone. Yes. Now coming to the second example, that is referred to as gene therapy. Gene therapy. Now suppose there is a person who is born with some hereditary disease. Okay. Suppose there is a child and this child is suffering from a disease by birth. Okay. This child has certain diseases from the birth. So these you can say problems, they can be corrected by using the gene therapy. So gene therapy हमारी वो टेक्निक है जिससे हम किसी बच्चे के अंदर जो हेरिडिटरी डिजीज़ है, जो डिजीज़ बाय बर्थ प्रेजेंट है, उन्हें भी करेक्ट कर सकते हैं. Okay, with the help of a technique which is called as gene therapy. So we can say gene therapy, it is a method. Okay, gene therapy is a method to correct a gene defect diagnosed in a child or an embryo. And here the genes, they will be inserted into the person's cells or tissues to correct that disease. And because of this when we insert the gene into the person, whether that person is, suppose initially the person is not having the gene. So by utilizing the gene therapy, when we induce that gene, we insert that gene into the cells or the tissues of that particular child, it will compensate for the non-functional gene that is present in the child. तो हमने क्या किया, एक बच्चा है, उस बच्चे के अंदर एक disease present है जो by birth present है, it is a hereditary disease. So what we have done, we have isolated the gene and we have inserted that gene into the child so that it can produce the functional gene and that functional gene will produce a desired protein in the child, and the disease of the child will be cured. So this is all about your gene therapy. Now, initially, the first clinical trial that was done regarding the gene therapy, it was done in 1990s on a 4-year child. There was a girl, and that girl was only 4 years old. And the initial trials, they were the first clinical trials of this gene therapy, they were conducted on this girl itself. Now imagine, suppose there is a gene. Okay, this is a gene. Now this gene basically codes for an enzyme ADA. ठीक है, एक जीन होती है, उस लेट्स सपोज दैट जीन है, उस जीन की वजह से एक एंजाइम प्रोड्यूस होता है. ओके, दैट इज कॉल्ड एज एडिनोसाइन deaminase. This enzyme is called as adenosine deaminase. And when this enzyme is present, okay, then your immune system will function properly. So immune system functions properly. Okay, everyone? So there is a gene. That gene will code for an enzyme which is called adenosine deaminase, and that enzyme is responsible for the normal functioning of the immune system. Now just imagine, this girl does not have this gene. ये जो बच्ची थी उसके अंदर ये जीन प्रेजेंट नहीं थी. Now because of this, the enzyme ADA was not produced. Okay, because there was some problem in this gene, so the enzyme ADA was not produced. So in this girl there was deficiency of adenosine deaminase enzyme. Okay, बच्ची के अंदर इस enzyme की कमी थी, because either the gene was not functioning or there was some problem with the gene, and because of that this ADA enzyme was not functioning properly, and because this ADA enzyme was not there, the immune system was not functioning. Okay, so this gene was not there. उस बच्ची के अंदर ये जीन प्रेजेंट नहीं थी, सो उसकी बॉडी में एडेनोसाइन डीएमिनेज एंजाइम भी प्रेजेंट नहीं था, और इसीलिए उसका इम्यून सिस्टम प्रॉपर्ली फंक्शन नहीं कर रहा था. ओके, क्लियर? नाउ in some children this ADA deficiency was treated by bone marrow transplantation, okay, or by using the enzyme replacement theory. कुछ बच्चों में ये जो प्रॉब्लम थी ADA डेफिशिएंसी की, ये बोन ट्रांसप्लांटेशन से या एंजाइम रिप्लेसमेंट थ्योरी से करेक्ट की गई थी. बट both these techniques they does not provide a permanent solution. Okay, clear? They does not provide any permanent solution. It was not that the disease was completely cured. Okay, that is why there was need that we should subject it to gene therapy. Now what we do in actually gene therapy, हम gene therapy में क्या करते हैं कि इंडिविजुअल के अंदर से लिम्फोसाइट्स को एक्सट्रैक्ट करते हैं. जो हमारा पेशेंट है, पेशेंट से उसके लिम्फोसाइट्स को एक्सट्रैक्ट करते हैं और एक कल्चर में ग्रो करते हैं. ओके, so suppose this is a person, this is a patient. So from the patient we will extract the lymphocytes and we will transfer it to a culture. So on the culture we will provide enough nutrients so that these lymphocytes they can grow very easily. Clear? Is this clear, everyone? Okay. Now what will we do? We will insert a functional ADA complementary DNA. ठीक है, अब इस कल्चर के अंदर हम क्या करेंगे, ADA यानी complementary DNA को इंड्यूस कर देंगे. Now this complementary DNA, it will go and it will bind with the human ADA. ठीक है, हमने क्या किया कि जो जीन रिस्पॉन्स जो फंक्शनल ADA था, दैट इज़ फंक्शनल एंजाइम के जो कॉम्प्लीमेंट्री DNA था, दैट वी हैव प्रोड्यूस यूजिंग अ रेट्रोवायरल वैक्टर. ठीक है, हमने एक रेट्रोवायरल वैक्टर लिया, उसके अंदर हमने ADA के कॉम्प्लीमेंट्री DNA सिंथेसाइज किया है. अब वो DNA हमने जो है इन सेल्स के अंदर इंजेक्ट कर दिया. नाउ दीज टू DNA, कॉम्प्लीमेंट्री DNA, दे विल बाइंड विद ईच अदर, एंड व्हेन दे विल बाइंड विद ईच अदर, दिस विल लीड, सो दिस जीन विल बी करेक्टेड. ADA deficiency will be corrected and these lymphocytes they will be injected back into the human body. अब ये जो lymphocytes हैं जिनके अंदर हमारी जीन प्रेजेंट है, जो जेनेटिकली इंजीनियर्ड ADA जीन है वो हमने इंसर्ट कर दी है लिम्फोसाइट्स में. अब इन सेल्स को वापस हम पेशेंट की बॉडी में इंड्यूस कर देंगे. इफ़ दिस प्रोसेस इज कैरिड आउट इन द अर्ली एम्ब्रियोनिक स्टेजेस, ये जो प्रोसेस है हमारी जीन थेरेपी का, सो दैट वी कैन करेक्ट द ADA डेफिशिएंसी. इफ़ दिस इज डन एट अ वेरी अर्ली एम्ब्रियोनिक स्टेज, देन दिस विल प्रोवाइड अ परमानेंट क्योर टू द पेशेंट. अब इसकी वजह से जो है वो उसकी डिजीज़ जो है वो परमानेंटली क्योर हो जाएगी. ओके, क्लियर? एज आई टोल्ड यू, फर्स्ट ऑफ ऑल हमने लिम्फोसाइट्स को एक्सट्रैक्ट किया, उसको कल्चर में ग्रोन किया. नाउ हमने जो ADA की जो जीन है उसको हमने यू कैन से we have extracted and we have prepared a complementary DNA, and then we inserted a complementary DNA into the lymphocytes. Then these two complementary DNAs, they will bind with each other, and there is a corrected ADA gene which is now present in these lymphocytes, and again these lymphocytes, they will be inserted back into the patient. And if this whole process is carried out in the early embryonic stages, then this particular gene therapy, it will provide a permanent cure for this disease, or permanent solution to this disease. Okay, so this is how exactly we perform the gene therapy. Clear, everyone? Now coming to the third part, which is called as molecular diagnosis. I hope you've heard this term, molecular diagnosis. See, when we want that a disease must be effectively corrected, so in that case we want that disease must be identified early. Okay, it should be identified early, and then secondly we should understand the pathophysiology of the particular you can say virus, particular pathogen which causes this disease. ठीक है, अगर हम चाहते हैं कि कोई बीमारी जल्दी से ठीक हो जाए तो सबसे पहले हम ये चाहेंगे कि वो पहले आइडेंटिफाई होनी चाहिए, एंड वंस इट इज़ आइडेंटिफाइड we should know which pathogen causes that particular disease. Okay, so initially we used to have the conventional methods. Starting में जो हम लेते थे वो हम conventional method use करते थे, and by using the conventional method it was not possible to diagnose the disease at a very early stage. Okay, so that is why we need the you can say latest techniques, that is your recombinant DNA technology, the PCR, and the ELISA technique, that is your enzyme linked immunosorbent assay. So these are the three techniques that help in the early identification of a disease. And once the disease is identified at an early stage, then there are chances that it can be cured. Okay, clear? And when we want to cure it, we need to understand which pathogen causes that disease. Now, as I told you, there are three techniques which can early identify the presence of a pathogen. That is your rDNA, that is recombinant DNA technology, PCR, and the ELISA. Now, the presence of the pathogen, it is normally when we identify whether pathogen is present in the body or not, then pathogen can be identified only when the body produces certain symptoms. देखिए, हम किसी भी पैथोजन को तभी आइडेंटिफाई कर सकते हैं जब वो हमारी बॉडी के अंदर सिम्टम्स या साइंस प्रोड्यूस करता है डिजीज़ के. ओके, और जब ये सिम्टम्स और साइंस प्रोड्यूस होते हैं हमारी बॉडी में, तब तक आपके अंदर पैथोजन की अमाउंट इतनी ज़्यादा इंक्रीज हो चुकी होती है. ओके, by this time the concentration of the pathogen has already increased in the body. Clear? Because our conventional methods, they cannot detect at a very early stage. Now, when we want to detect at a very early stage, that means a detection should be done when the number of pathogens is low. So, अगर pathogens का number slow है और उस stage में हम उसे identify कर लेते हैं, then disease को बहुत आसानी से cure किया जा सकता है. Now, this identification, when the pathogens are present at a very low concentration, it is done by the PCR technique. So, PCR technique, that is polymerase chain reaction, the amplification process, it will help in identification of the pathogen at a very early stage and when its number is very, very low. Okay. So let's see where is PCR technique used. First of all, the PCR, it is used to detect the HIV, that is the virus that causes the AIDS. So first of all, it is used to identify the HIV virus, whether it is absent or present in the human body. Secondly, it can also detect the mutations in genes that can cause cancers. Thirdly, it can identify the genetic disorders. So PCR technique, it is very, very useful in identification of the AIDS, in identification of the mutations that may lead to cancer, and also in the identification of certain genetic disorders. Now, how basically PCR is performed? Now, in PCR, what we do, we take a single stranded DNA. हम PCR में क्या करते हैं, सिंगल स्ट्रैंडेड DNA और RNA को यूज़ करते हैं, और इस DNA और RNA को हम टैग कर देते हैं बाय यूजिंग अ रेडियो एक्टिव प्रॉब. सो व्हाट विल डू, वी विल हैव द DNA और द RNA. ओके, हमने DNA और RNA लिया है और हमने इस RNA DNA को टैग कर दिया है. ठीक है, वी हैव टैग्ड इट बाय यूजिंग a radioactive molecule. So now this DNA, it will behave like a probe. Okay, now this probe, we have tagged this DNA, this will behave like a probe. Now this probe, it is hybridized with the complementary DNA. अब ये जो probe है हमारा, इसे हम complementary DNA के साथ hybridize कर देंगे. Okay, we will hybridize it with its complementary DNA which is present in the cells. And then we will perform the autoradiography technique. तो सबसे पहले हमने क्या किया, हमने DNA और RNA को टैग कर दिया by using a radioactive molecule, and this DNA molecule will act as a probe. Now this probe, we have allowed it to hybridize with this complementary DNA which is present in the clones, and this DNA will then be detected by using the autoradiography. Okay, the clones that have mutated genes, okay, the clones that have mutated genes will not appear on the photographic film because they do not have the probes. Okay, जिन cells के अंदर ये probe absent होगा जो हमने insert किया है, वो रेडियो ऑटो रेडियोग्राफी से डिटेक्ट नहीं होंगे, और जिनके अंदर ये probe present होगा, they could easily be detected on the photographic film. Okay, so this is how we can identify this is how we can identify, or we can use the PCR technique, right? Next comes the last technique, which is called ELISA. That is, what do we call it as? Enzyme linked sorbent assay. Okay, now this basically is based on the antigen antibody interaction. देखिए, जब हमारी बॉडी के अंदर कोई भी पैथोजन मूव करता है, अंदर चला जाता है, so that starts behaving like an antigen. Now, in response to the pathogen that has entered our body will produce certain proteins which are referred to as antibodies. तो जब ये पैथोजन हमारी बॉडी में जाता है, ये जाकर एक एंटीबॉडी के साथ बाइंड हो जाता है. सो दिस मीन्स इट ट्रिगर्स एन इम्यून रिस्पॉन्स. ओके, ठीक है, समझ आ गया? तो हमारी बॉडी के अंदर एंटीजन्स हैं. एंटीजन्स मीन्स जो फॉरेन पार्टिकल्स हैं जो हमारी बॉडी के अंदर जाते हैं और हमारी बॉडी के इम्यून सिस्टम को एक्टिव कर देते हैं. So these antigens, they will bind with the antibody and they will initiate a immune response, right? Clear? Now what will happen? Now because of this binding, there is secondary antibody will be produced. This secondary body will activate the enzymes, and these enzymes, they will convert the substrate into products. ठीक है? समझ आ रहा है प्रोसेस? एंटीजन है, एंटीजन के साथ आपका एंटीजन जो है वो आपकी एंटीबॉडी के साथ बाइंड हो जाएगा. इसकी वजह से आपका इम्यून रिस्पॉन्स ट्रिगर हो जाएगा. सेकेंड एंटीबॉडी जनरेट होंगी. ये सेकेंड एंटीबॉडी जो है एंजाइम्स को एक्टिवेट करेंगी और ये आपके सबस्ट्रेट को प्रोडक्ट में कन्वर्ट कर देंगी. क्लियर? ओके, सो नाउ व्हाट विल वी डू इन एलिसा. एलिसा में हम क्या करेंगे कि सपोज हमने एक सैंपल इंजेक्ट किया है, ठीक है. इस सैंपल को जब हम इंजेक्ट करेंगे, ये आपकी एंटीबॉडी के साथ बाइंड हो जाएगा. सो फर्स्ट पार्ट इज बाइंडिंग. ओके, नाउ दिस विल बाइंड. Now second phase, what will we do? We will try to remove the antibodies, and we will see the formation of secondary antibodies. Secondary antibodies अगर बनती हैं, एंजाइम इनिशिएट हो जाएगा और एंजाइम से प्रोडक्ट की फॉर्म में कन्वर्जन हो जाएगा. तो अब हम इस प्रोडक्ट को डिटेक्ट करेंगे. ठीक है, we will detect the product that has been formed by this enzyme. ठीक है. एंटीजन एंटीबॉडी पे बाइंड किया, उसकी वजह से सेकेंडरी एंटीबॉडी का प्रोडक्शन बढ़ा, सेकेंडरी एंटीबॉडी ने एंजाइम को एक्टिवेट किया और वो सबस्ट्रेट को प्रोडक्ट में कन्वर्ट करेगा। अब एलिजा टेक्निक क्या करती है कि इस प्रोडक्ट को आइडेंटिफाई करती है, द प्रोडक्ट। नाउ दिस प्रोडक्ट विल बी डिटेक्टेड by using this technique. Okay, or we can easily detect the antibodies that have been synthesized against this pathogen. या हम ये डिटेक्ट कर सकते हैं कि हमारी बॉडी के अंदर कितनी एंटीबॉडीज का कॉन्सेंट्रेशन प्रेजेंट है अगेंस्ट दिस पैथोजन. इफ दिस पैथोजन इज प्रेजेंट देन द कॉन्सेंट्रेशन ऑफ द एंटीबॉडीज प्रोड्यूस्ड इन द बॉडी विल बी मोर. Okay, so by this technique we can detect whether a particular pathogen has entered and a particular protein is present in our body. If that protein is present, this means this particular pathogen is present inside the body. तो हम एंटीबॉडी की कॉन्सेंट्रेशन से ये पता लगा सकते हैं कि कौन सा एंजाइम या कौन सा पैथोजन है जो हमारी बॉडी के अंदर यू कैन से प्रेजेंट है. And again we can use a desired technique to eliminate that pathogen later on. Okay, so all these three techniques, that is the recombinant DNA technology, PCR and ELISA, they help in the early detection of the you can say disease causing pathogens at a very early stage, and they make it possible that the diseases can be cured at the initial stages itself. Okay, I hope there is no doubt. If there is any doubt, you can ask dear students. Right. So let's come to your homework for today. That is, you will be making notes of this topic and you will be reading the page numbers 210 to 212. And if there is any query, any doubt, anything that you feel you can discuss, you can send your queries to me personally. Okay? Okay, so that's all for today. Bye everyone.